Hello and welcome everybody to my channel Omni Bioentrance, and today we will be discussing GATE Life Science question paper conducted in the year 2024. I will be discussing the biochemistry, biotechnology, cell biology, plant physiology, molecular biology, genetics and immunology questions. So coming to the first question. An enzyme catalyzed conversion of a substrate at 298 Kelvin proceeds by Michaelis-Menten mechanism. The Lean-Beaver work plot for the analysis of the experimental data has the intercept along the y-axis of 0.357 millimoles per decimeter per second and a slope of 2.10 second. The correct Michaelis-Menten constant for the reaction is. So they have asked the Km value. So to calculate the Km value, you have the equation slope by intercept. So substitute the values, you will get 5.88 millimoles. So mole will go on the top, mole raised to minus 1 here will go on top. So it will be mole, and dm cube will become dm minus 3. So the answer here is option A, 5.88 millimole dm raised to minus 3. Okay. The spectrum of protein obtained using electron spray ionization mass spectrometry is shown below. Two peaks, one of MZ is 2960.6 and the other M by Z is 3552.5 are marked. So this is that peak and this is the other peak. The mass of the protein associated with the M by Z 2960.6 peak is. So here if you see, we know that two adjacent peaks, they differ by one charge. Okay, ONE, 1 charge. Remember that. So you have the formula for calculating molecular mass. M by Z is equal to molecular weight plus NH plus by N. So M1 by Z is equal to molecular weight into NH plus by N, and M2 by Z will be molecular weight plus N plus 1, because two adjacent peaks differ by one charge. So it will be N plus 1 into H by N plus 1. 1 minus 2. So this is the simplification for M1 minus 2, and then you put in the values. So N into 355.5 minus 2960.6 will give you N as 5. So substitute the value of N is equal to 5 in equation 1, that is this. You will get the molecular weight, that is 17,757.46 Daltons. Then next one. Which of the following selectable marker genes is or are used for herbicide tolerance? They have asked herbicide tolerance during genetic transformation of plants. HPT, it is hygromycin phosphotransferase. It is not used in herbicide tolerance. It is for hygromycin resistance. BAR is phosphinothricin acetyltransferase, and it confers tolerance to phosphinothricin and bialophos herbicides. So it is a herbicide tolerance gene. NPT2 is for neomycin phosphotransferase and is used to, for kanamycin or neomycin resistance. PMI is phosphomannose isomerase. It is also not used for herbicide tolerance but for mannose selection. So the answer here is BAR. Then next one. In Calvin-Benson cycle, to produce one molecule of glyceraldehyde 3-phosphate by fixing 3 molecules of carbon dioxide, 9 molecules of ATP and dash molecules of NADPH are typically used. So 9 molecules of ATP and 6 molecules of NADPH will be used in Calvin-Benson cycle. Then next one. Match the inhibitors in group 1 with their respective targets in group 2. So oligomycin. Oligomycin is for ATP synthase. So P4. P4, okay, B we can see, and D, A and C we need not take into consideration. Antimycin A. Antimycin A is cytochrome BC1 complex. Q1. Q1 is correct in B. So B must be the answer. Okay, we will see. Then DCMU. DCMU is for photosystem 2. And valinomycin is for K plus ionophore. So P4, Q1, R2, S3 is the answer. Then next one. With reference to Agrobacterium tumefaciens mediated plant transformation, match the virulence factors in group 1 with the protein types in group 2. So VIRG, it is a transcriptional activator. So P3. Okay, B and C, D we have to see, A and C we need not see. Then VIR-A. VIR-A is a kinase. So Q1. VIR-E, it is single-stranded binding protein R4, and VIR-C, it is a helicase. So the answer here is option D, that is P3, Q1, R4 and S2. Then next one. In a genetic cross between two pure line parents differing in the two independently segregating traits, that is plant height and flower color, all the F1 plants were tall with purple flowers. So F1 is tall with purple means tall is the dominant trait and purple is the dominant trait. Dwarf and white are recessive traits. In a test cross population involving these F1 individuals, the expected percentage of dwarf plants with the purple flower would be. So you know, test cross, it usually gives the 1:1:1 ratio, and all will be phenotypically and genotypically different. So for dwarf plant with purple flower, you will get 1/4, that is 25%. So I have done the genetic cross over here. So the parent will be, they have told, pure line parents. So capital T, capital T, capital P, capital P. P is for the purple color and T is for tallness. This small t is for dwarf and small p is for white. So F1 will be TTPP. Then you cross it with the homozygous recessive parent, that is your test cross, and you will get 1:1:1 ratio of, that means the dwarf plant with purple flower will be 25%. Then next one. The mRNA of a hypothetical gene HSDU is 800 nucleotide long and encodes a protein of 160 amino acid residues. The calculated length of HSDU CDS, CDS is coding DNA sequence, would be how many nucleotides? So each amino acid, you all know, it is encoded by 3 nucleotides. So the length of CDS will be number of amino acid residues into 3. So it will be 480 nucleotides. That is 160 into 3, that is 480 nucleotides. Then next one. The most abundant class of immunoglobulin in serum is, it is IgG, which is the most abundant. Then next one. Which of the following double-stranded sequence will not be recognized by type 2p restriction endonucleases? So type 2, they recognize sequences which are palindromic, that is they will read the same in 5', 3' direction in both the strands, that is in this direction also as well as in this direction also. So GGTACC, in 5' 3' direction in the top strand. So in the complementary strand also it should be GGTACC when read in 5' to 3' direction. So bottom strand it is GGATCC, that is 5' 3' direction. So it is palindromic. It will be recognized by type 2 restriction endonuclease. Similarly, B also will be recognized. It is GGATCC. GGATCC. It will be recognized. CATATG. CATATG. So C also will be recognized. But if you see D, it is GATTTC. And in 5' 3' direction in the complementary strand is GAAATC. It is not the same. So D will not be recognized by the type 2 restriction endonuclease. Then next one. Which of the following events occur during translation in prokaryotes? tRNA binding to the start codon of the mRNA on the 30S subunit of the ribosome. That is correct. Anticodon of tRNA binding to the start codon of the mRNA on the 50S subunit of the ribosome. It is not 50S subunit, it is 30S subunit. So B is not correct. The ribosome continues to move along the mRNA to add new amino acids to the polypeptide. That is correct. The polypeptide is released when the ribosome reaches the stop codon. That is also correct. So A, C, D is the answer. Then next one. Which of the following is or are consequence of nitrous acid mediated deamination? So deamination of cytosine, adenine, all these will give rise to, I will explain. So deamination of cytosine results in uracil. Uracil which pairs with adenine. So when the next round of replication, the GC will be converted to AT, or you will get GC-AT transitions. Then deamination of adenine will give rise to hypoxanthine, and this hypoxanthine it will pair with cytosine. So in the next round of DNA replication, you will get AT to GC transition. So the consequence of nitrous acid mediated deamination is deamination of cytosine, adenine, guanine, GC to AT transition and AT to GC transition. Addition of alkyl group to the bases, it is alkylation. It is not like methylation and all. So it is not deamination. So D is not a correct statement. Then next one. Which of the following is or are true about the Escherichia coli chromosome? It is typically bound by histones. No, E. coli doesn't have histones. It is circular in nature. Yes. It is found in the nucleoid. Yes. It contains multiple origin of replication. It contains only single origin of replication. So that is also not correct. Then next one. Which one of the following statements about gene expression is incorrect? DNA is transcribed to mRNA. That is correct. mRNA can be reverse transcribed to DNA. Of course. mRNA can be translated to proteins. Yes. Proteins can be reverse translated to mRNA. That is cannot take place as per the central dogma of molecular biology, which states that genetic information flow from DNA to RNA to proteins, and once proteins are synthesized, it cannot be converted back to mRNA. So D is the correct answer. Then codon bias is correlated with the relative frequency of which one of the following types of RNA? So codon bias, it is the I can tell you that their genetic code is degenerate. That is, one amino acid can be coded by different codons, but the cell of one particular organism will have a bias towards one of the codons out of the many which code for the same amino acid. Okay, for example, when I tell you the example, you will understand. So if you take leucine into consideration, it has 6 different codons, and out of these 6 codons, one is CUG. So in humans, the codon CUG is more frequently used than the other 5 codons which encode leucine. This is codon bias. Okay. And one reason for this bias is the tRNA molecule that recognizes the CUG codon, and it is more abundant in the human than the tRNA molecule that recognize other leucine codons. That is codon bias. So they have asked here, codon bias is correlated with the relative frequencies of which one of the following type of RNA. So it is the frequency of the tRNA. So answer here is option D. Then next one, CREB1 is a eukaryotic transcription factor. In which one of the following compartments of the cell is CREB1 located? So it is located in the nucleus. CREB1 is cyclic AMP response element binding protein 1. Then next one, the human genetic code is triplet in nature with 64 codons made using 4 nucleotides. So 4 raised to 3 will give you 64 codons. Now if the human genetic code has doublet in nature, the number of codons theoretically possible from 4 nucleotides. So 4 cube gave you 64 means, now when the genetic code is doublet, that is 2 will be, nucleotides will be coding for one amino acid, then number of codons theoretically possible from 4 nucleotides will be, so 4 raised to 2, that is 4 into 4, that is 16. So the answer here is 16. Then next one, which one of the following statement is not true of glycosaminoglycans? So glycosaminoglycans are composed of repeating disaccharide units. That's correct. Glycosaminoglycans consist of amino sugars that are frequently sulfated. Yes. Hyaluronic acid is an example of glycosaminoglycan. That's also correct. Methionine is the predominant amino acid to which glycosaminoglycan chains are conjugated to form proteoglycans. It is not methionine, it is serine. So D is the incorrect statement. Then next one. Consider the following nucleotide sequence. Assume canonical initiation, canonical termination, no post-translational modification, and the average molecular mass of amino acid to be 110 Daltons. The theoretical molecular mass of the polypeptide translated from the above nucleotide sequences. So where is AUG? AUG is here. We have to see how many amino acids are produced from this nucleotide sequence. So 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14. UAG is a stop codon. UAG is a stop codon. So 14 amino acids are produced, and molecular mass of one amino acid is 110 Dalton. So the molecular mass of 14 amino acid will be multiplied by 110, that will give you 1540. Then next one. The pKa of a buffer solution with pH 5 consisting of 0.4 molar sodium acetate and 0.04 molar acetic acid is. So this is by Henderson-Hasselbalch equation, and we know pH is equal to pKa plus log concentration of A by concentration of HA. So substitute the values. pH is given as 5, pKa we have to find out, then log 0.4 by 0.04. Rearrange the equation to calculate pKa, we will get it as 4. So thank you all for watching my channel. Please do like, share and subscribe. Thank you.